Let's get started. Will somebody be my timer? I mean, I went, I went like way over a couple days ago. That was on purpose. And then on whatever it was, let's say Wednesday, on Monday, I went over by accident. So will somebody be my timer and we'll cut class like when it actually ends? We'll stop. But for the sake of today, the endocrinology stuff and cell signaling cascades. This is really what we're going to get into with mTORs is these cell signaling cascades. mTORs is critical regulator of metabolism, and it is a cell signaling cascade. mTOR itself, mammalian target of rapamycin. mTOR is an enzyme. It's a kinase, and so it has upstream activators and downstream targets, and so it's phosphorylating stuff, and we're going to talk about all of that and regulation of all of these things. It's a bunch of signaling cascades. So I've been talking about cell signaling, whether it's mechanotransduction or just whatever phosphorylation cascades for a while. If it's lipolysis or glycogenolysis, what is adipose triglyceride lipase doing? Well, it's in lipolysis, which is just a bunch of cell signaling to mobilize fat. And so cell signaling has been a theme for quite a while. Even here, right? PKA, protein kinase A. We're going to talk a lot about PKB today, protein kinase B, and its sort of panoramic qualities, pan kinase of a kind. And adenylyl cyclase and protein kinase A, just these cell signaling cascades, these things have been coming up for a really long time. Insulin binds to its receptor, IR, insulin receptor substrate, right? And you get to PI3K kinase. And then, PKB, protein kinase B, or AKT, and we're mobilizing GLUT4. It's just cell signaling cascades. And we'll go into a little bit more detail in this and what's upstream and what's downstream from PKB and sort of side streams of PKB. But the anabolic hormones, if it's not, androgens and estrogen, and with the catabolic hormones, if it's not cortisol, when we're getting into all of these polypeptide hormones, amine and peptide protein hormones, amino acid things, it's a bunch of signaling cascades, thyroid aside. But even with the thyroid hormones, and even with estrogen and even with testosterone, you're seeing cell signaling you're seeing mTOR signaling with those things, PI3K, PKB, mTOR. So this stuff, that stuff, nutritional effects. These things are synthesized from cholesterol, so you know what diet can do to it. Estrogen, the difference between that and androgens in terms of their anabolism, their growth. Myostatin versus follistatin. IGF, insulin-like growth factor, downstream from growth hormone. IGF there's going to be a lot of PI3K, PKB signaling coming from IGF, and growth hormone and what it is and what causes it to be promoted, what causes it to be inhibited, and exercise and sleep and strenuous exercise takes a little bit of time. And strenuous exercise, the thyroid stuff that's sort of fascinating as this mimicking of steroid hormones, but they're actually amine hormones. Uh, they're from one amino acid, so in that polypeptide class, but the amine hormones. And AKT, that's PKB, mTOR, P70S6K, the thyroid hormone stimulates, anabolism, protein synthesis in this way. That is one of its actions. The arguments against steroids, and you get to keep them if you want. Whatever you want to believe. I'm not here to tell you what to believe. I'm just here to say I've heard arguments that make no sense, and there may be additional arguments that people haven't made before that do make sense, and I'd be happy to hear them. But usually, what it comes down to, you listen to somebody long enough, and this is the point that they keep hammering without knowing it. And even in all of those publications that were coming out, everything from ESPN to the FDA, that everyone was just talking about home runs. People just cared about sports. That's really all anybody cared about. There's no shortcuts. This is cheating. Everyone's getting something for nothing, and that's all anyone ever talks about in the end. baseball great Jim Bunning. There are no shortcuts in life. So, with anabolic androgenic steroids, exogenous steroids, taking them as drugs, just be able to answer some of these questions about them. What does it look like on the liver? What does it look like on the heart? The actual science of it, and metabolism. So we introduced last lecture was just a ton of enzymes. It was let's just hit metabolism hard in one day, just a lot of metabolic punches and then leave. And so we went over those 8 different versions of enzymatic alteration, behavior alteration, mostly inhibition. So we have allosteric modulation. You can increase the activity or decrease it, the attraction with its substrates, its reactants or its substrates. If you bind to an allosteric site, you can change the shape of the active site, and that can facilitate or inhibit binding with its substrate. There's competitive inhibition, which is the substrate and the the enzyme. Um, the substrate has a competitor, right, that can also bind to the active site. It doesn't bind to an allosteric site. It can bind to an active site. And if it binds to that active site, there's only one hole there, right? There's only one divot, one socket. And so only one plug can go in that one socket, and the inhibitor got there first. So that's competitive inhibition. Suicide is similar, but it's an irreversible version where the inhibitor is derived from the substrate, and then it starts to undergo the chemical change, and then this isn't what I thought it was. Non-competitive inhibition, it binds whether the substrate's bound, doesn't care. It's not competitive. It doesn't, you're bound, you're complex with your substrate. I don't care, I'll bind. Are you free? I will bind. It really doesn't care. And it just affects the reaction. It affects the activity. There's uncompetitive. That's the one that is potentiated by binding with the substrate. So uncompetitive, if you see that word potentiated by, it's going to be uncompetitive, where its socket doesn't open up until the inhibitor, or until the substrate has bound. So the socket for the inhibitor will open up after the substrate has already bound. There's mixed, which is like non with a preference. I'm not going to ask about that one, but you may encounter it in the literature if you go in the direction of studying metabolism or studying drugs. Pharmacology, I'm sure it'll make a bunch of appearances, right? This is just sort of chemistry of metabolic interactions, and then let's mess up some enzymes, and how do we do that? So mixed you may encounter, but I'm not going to test you on it. This is sort of a little bit of a weird one. Accumulation of product. That one is, we talked about hexokinase can be backwardly inhibited with the accumulation of G6P, glucose 6-phosphate. You get G6P from glycogen or from glucose. If you're going to get it from glucose, you go through hexokinase. If you get from glycogen, you go through that glycogen phosphorylation and then phosphoglucomutase to get to G6P. And that will actually become important later when we're talking about the burning sensation and stuff like that, and ATP accumulation, and how lactate does not cause a burning sensation, all that stuff. Well, that's later. I'm not going to test you on it. But if you accumulate a bunch of the product, there's no reason for that enzyme to keep doing its thing. You have a ton of the what comes out of it, right? You already have the product. We don't need the substrates to keep pouring into this thing. We already have the product, so you backwardly inhibit it. And then I think that's all of them, other than phosphorylation, if I missed one. But phosphorylation, metabolism is largely a bunch of phosphorylation cascades. You attach a phosphate to something, and it changes its behavior. Either it turns it on, turns it off, something like that. Okay, I have changed the background. I have changed the appearance. We're moving into mTOR mode. So we're changing. We're putting on big boy clothes. right? To get into mTOR signaling. This is like a totally different section now. And we're just going to draw from everything we've learned all semester. But we have a new, we're just going to plow and plow and plow and just move forward, move forward, move forward. And I will repeat points as we go, but I'm going to pick up where we left off in mTOR for the next couple of lectures. This one, the next one, and the next one is just going to be novel, novel, novel. I'm not going to repeat a bunch of prior information. I will rephrase some stuff and remind you of stuff as we move forward. But this is what we're gonna cover, and not just today. This is what we're gonna cover in the next few lectures. So, what's hypertrophy? Like, what is it, right? And then, what does hypertrophy is happens? I don't remember what I was getting at there. Um, what causes hypertrophy to happen? What results of hypertrophy are there? So, what is hypertrophy? What makes it happen? And what are the consequences of hypertrophy and how it occurs? It's going to be mTOR mostly mTOR. And there are other mechanisms of hypertrophy, MAPK or testosterone's direct effects. So there are other mechanisms of muscle growth, but mTOR is this critical regulator of skeletal muscle. And then we're going to end this little block with combining mTOR, what we're learning about now, with everything else we've learned. Henneman's size principle, specificity of adaptation, and a lot of these previous concepts we've been talking about buddy up with mTOR in a interesting way, in a prescriptive way, in a way that you can use on your dinner plate, on a way that you can use in the weight room, it's stuff that you can incorporate into your own life. But we will start combining a lot of what has come before with mTOR. So let's start with what hypertrophy is. Muscle metabolism, right? So muscles are constantly growing, muscles are constantly shrinking. It's just a bunch of proteins in there. There are a bunch of a muscle cell is loaded with proteins. It's first, like, skeletal muscle is probably, it depends on the person, maybe half of your body mass. Again, this depends. This is a big give or take, but a lot of a lot of your corporeal bulk is muscle, and muscle is loaded with proteins. When I go to Uganda, it's sort of famous for its birds, and it's like they do not withhold the marketing of Uganda as this destination where 50% of all of Africa's birds, of the variety of species, are in Uganda. And maybe 10%, somewhere around there, one out of every 10 species of birds in the world is in the whole world over is in Uganda. Muscle is a little bit like that with its proteins. 70% or so of the proteins in your body are native to your muscle. Now, most of them exist elsewhere also, but it is this home. If you were to take the magic school bus on a protein safari, skeletal muscle, that's where you want to go. That's where you're going to see this amazing diversity. I mean, so many different proteins are expressed there. And the protein turnover, this, we're synthesizing new proteins, we're degrading old proteins. This is constant, and it can be an anabolic process where, if hypertrophy is just synthesis outpacing degradation. If you're breaking down your proteins slower than you're building them up, that's hypertrophy. Atrophy is just degradation outpacing synthesis. So hypertrophy and atrophy are just a matter of protein turnover, of muscle metabolism. And we are constantly doing this to try to be more fit, right? To try to be compatible with our environment, right? To try to be more suitable to the environmental stresses, to be apt or appropriate to whatever we're experiencing. And there are just, again, so many. It's not just actin and myosin. Right? We've talked about a bunch of stuff. We talked about titin and nebulin and alpha actinin and desmin and I mean, there's all of these different proteins that are going to, myomycin, M protein, all of these different proteins that are being deposited in here. And so it's not just like actinomycin, right? There's a ton of stuff. It's this that the safari of protein has such diverse wildlife that is native, that is expressed there. So why does hypertrophy happen? Just the questions like, why is exercise good for you? Okay, why does resistance training, why does this cause hypertrophy? Now, why and how, two totally different questions. Okay, why questions are these have purpose to them. Why did you do that? I hated the person, so I slashed their tires or whatever. There's purpose, not how did you do it? Well, I sharpened my knife, and so when little kids ask questions like, why is the sky blue? That's not a good question. And I think it's fair to say that to the kid. We got to educate kids earlier, right? Why is the sky blue? Bad question. Come up with something else because that doesn't mean anything. Why is the sky blue? Well, Lord Almighty on the second day decided, man, blue, what a lovely color. The color of, Granny, Almighty Granny's eyes. And I'm feeling sentimental today. Let's make it. Why is purpose? And in physiology, purpose almost never has a place. It doesn't really mean anything. How is the sky blue? How questions are mechanisms. And how questions like, well, our perception of color is the interpretation of way. I'm not a physicist, so you correct me if you can. Our perception of different colors, the blues and hues and purples and whatever. uh, this is perception of interpretation of wavelengths. And up in the upper air, we have these gases, we have oxygen, we have nitrogen, and the wavelengths scatter, and these really short blue wavelengths scatter into perception. All right, so that's whatever I learned. And that's a how answer to a how question. Now, we're starting with why. Why does hypertrophy happen? Well, exercise is horrible for you. Okay, exercise is a really threatening endeavor. So just take these two people, these two cartoons, let's say they're real people. This person is clearly exercising very hard. He is warping the wind, he is distorting the air, and like sonic booms are following him and whatever. I mean, this person is exercising very hard. This person is leisurely strolling in warm-up pants and a scrubs top. This person and cocaine eyes and like a psychopath serial killer smile. This person is not at any risk of cardiovascular damage. This person is not going to have any type of infarction, right? There's not going to be, you're not going to be throwing clots around anywhere. No stroke on this body, right? This is not threatening. This person is putting their body at enormous risk. This is lethal. This is like, look at that face. That's lethal to other people. This is lethal to himself. And the reason we adapt to exercise is because exercise threatens us. Exercise is good for us. I said this on like the first, maybe second lecture. Exercise is good for us because it's bad for us. It's what I coined the positive injury principle, and I'm sure there's a better way to characterize that. But exercise is helpful for us because it threatens us, and our body re-evaluates its status. It starts reinforcing its tissues, rebuilding its structures stronger. And when we're in the specificity of adaptation section, I was talking about how if there's a flood in Stockton, that doesn't happen very often. Usually there's a drought, right? And so we have ways, I mean, everyone's installing it's like solar panels on their roofs because it's just nothing but sunlight all year round. But if there's a flood, you're just going to get sandbags. You're not really going to get. But if there's a flood every year, if there's 3-4 months a year, there's flooding. Now we start to take on specific adaptations to our homes for flooding, some retention wall or whatever it is. We dig a big empty moat or something and have it fill up with water and crocodiles or whatever. And but that is specificity of adaptation. in response to stress, in response to threats, in response to loads. That's why hypertrophy happens, is because the structural integrity of our tissues is not ready to withstand heavy loads, intense activity. And so high threats, high loads, high stresses, these things will induce adaptation. And one of the adaptations that reinforces skeletal muscle is hypertrophy. Let's make this thing bigger and more capable. Now, it's all geared at fitness, it is all aimed at um, survival, right? At self preservation, at fitness. And to do these things, it's a matter of compatibility with your environment. So, like an Aries is not compatible with another Aries, apparently, but is compatible with a Leo and is compatible with a Libra. And so this is the idea of like compatibility, right? If you're a Gemini, hook up with an Aries, but not a Taurus. I think because that would be bestial because a Taurus is like an animal. But uh, but this idea of compatibility in real life, not in like birth month. If it's hot out all the time, you're going to develop these adaptations that make you more heat tolerant. You're sweating and your um, vascular responses and you're going to have all these adaptations that are going to make you more compatible with a hot environment. So hypertrophy is to make you more compatible, more optimized, more appropriate to, more suitable with whatever an environment of a bunch of heavy loads. That's why hypertrophy happens. Specificity of adaptation, fitness, compatibility. That's why hypertrophy happens. How does it happen? How does hypertrophy happen? It's a bunch of cell signaling cascades. This is actually a good diagram. It just looks like Peanuts characters, like it's like Charlie Brown detail. And or no ligand, right? There's no signaling cascade happening. Ligand, this thing, a thing like a hormone or whatever, binds to a cell surface receptor and it activates that receptor. And then the receptor activates protein at the membrane, right? It's like a G protein coupled receptor, something like that. Some subunit of that G protein binds to that looks like adenylate cyclase or something like that. So we bind a ligand. We bind it's prostaglandins. It's some cytokine. It's insulin. It's glucagon. It's growth hormone. Whatever it is, a thing binds to a receptor, turns on the receptor, and then you activate some protein, and then protein at membrane activates protein in cytosol. And you just sort of start moving inward and inward and inward, and eventually you have your final response. Right now, often this is going to end at the nucleus, but it doesn't have to. GLUT4 is not living in the nucleus. I mean, so it ends at some specific intracellular response. So how does it occur? I usually describe it as a relay race, right? It's like a 4x1 relay race. You have 400 meters and there's four runners. Now there's going to be usually more runners than that in a signaling cascade, but you're not, I don't know of like a 10K relay race that everyone runs 100 meters. Be fascinating for a minute, but I don't think that sport would last in the Olympics. That would be a temporary appearance like the octopus wrestling. So, but if you look at what's happening here, we have a baton, right? This is a phosphate. For cell signaling, they're phosphorylation cascades. You phosphorylate something. Remember, if I phosphorylate my light switch, I turn it on or I turn it off. And if you phosphorylate some protein, you alter its behavior, activate or deactivate it. You don't have to flash like gang signs at each other. But so this runner in this relay race, you run, you run, you run, and then you're holding the baton. You're activated. You're phosphorylated. The baton is attached to you. You run, you run, you run, and then you give the baton to the next runner. Now you are no longer activated, probably, and the next runner. I mean, you can go do whatever you want. It doesn't matter. The next runner is active. That runner can't be activated until they're phosphorylated, until they're batoned. Okay, until this protein, this kinase, a kinase is a phosphorylating enzyme, protein until this kinase phosphorylates this protein. This protein's not active, or maybe it is active, and that's what shuts it down or whatever. But it changes its behavior. So this kinase phosphorylates this kinase, let's say, which phosphorylates another one and activates that one, and phosphorylates another one, activates that one. Eventually, it crosses a finish line, right? Eventually, that relay race, that signaling cascade, that cellular signaling cascade ends. It crosses the finish line. It's over. Yay! They get their trophy. And here's one of very few puns I'll ever use in my life. It's hypertrophy, right? In the case of mTOR, it's hypertrophy, where it's let's translate some proteins. Let's link together a bunch of amino acids, translate some proteins, and let's favor anabolism. Let's favor hypertrophy. In our protein turnover, let's get synthesis outpacing degradation. That's what this particular signaling cascade is going to do, is phosphorylate, phosphorylate, phosphorylate. Now, it's not all phosphorylation. There's other things that are occurring that are causing this cascade to keep marching forward to that finish line. But phosphorylation is really the big picture. mTOR is a kinase. It phosphorylates stuff. PKB, a kinase. It phosphorylates stuff. All of these things we're talking about. You have PI3K and you have AMPK and all of these. The bulk of these things we've been talking about are a bunch of kinases. They phosphorylate stuff. So, how does resistance training cause hypertrophy? It's mostly mTOR. And there's a couple of complexes. We're going to focus on complex one. We're going to go over complex two, but complex one, this is your hypertrophy. This is like you're growing in the weight room. Why am I not getting bigger in the weight room? I've been working out for however long and my voice is happy. That's it's probably like mTOR issues, and you can fix it. You're just doing it wrong. Now, maybe you're 90% type 1 fibers, and those fibers are less sensitive to mTOR signaling. Like they have a lower growth ceiling, and mTOR isn't going to be kind of revving its engine in type 1 fibers as it does in type 2. Okay, maybe there's genetic differences, but there's also modifications that people can make to their resistance training programs to make it more effective, to actually turn on mTOR, whether that's behaviors in the weight room or behaviors, cardiovascular activity or nutritional supplement types of activity. So it's mostly a bunch of mTOR signaling is what's happening. So what are some specific things that this molecule could be, right? And it's just, again, signaling cascades where we have relay molecules, a transduction. So if I say like mechanotransduction, well, over here we probably have some sort of transmembrane protein like an integrin. So we have an integrin relaying transduction inside of the mechanotransduction. Let's sense extracellular, that extracellular matrix, the loads going on outside of the cell, send that information into the interior and relay it, be like focal adhesion kinase, FAK, and just keep relaying that into the interior until you eventually have your finish line, you have your response. And again, mTOR signaling, our response is protein synthesis. It's let's make this bigger. Let's make this cell huge. Let's Ronnie Colemanize some cells. So what could a signaling molecule be, though? Again, let's say it's epinephrine. Okay, well, that's catabolic. Okay, that's PKA activation. If it is IGF or insulin, something like that, that's anabolic. Now we got PKB signaling. So there's a bunch of different things that it could be. Adrenaline is and epinephrine, those are the exact same thing. Adrenal, epinephrine. I mean, these are the same root words, just the top of the kidney, right? Insulin, right? So this one is going to be a PKA activator. It has its beta-adrenergic receptors and insulin receptors. This is PKB activation, growth hormone receptors. This is going to be JAK-STAT. So, Janus kinase, signal transducer activator of transcription. This is slightly different signaling cascade. Downstream from growth hormone, we're getting a bunch of IGF. So any of these things or lots more, but remember, all of these have to be taken by injection. If you eat them, they're just protein. Ham tastes better. Just eat your ham. There's just a bunch of amino acids in both. So signaling cascades, again, we've encountered these ones before. It's really all we're talking about, but we're going to get very anabolic. What we've been talking about is catabolic stuff. So, glucagon or epinephrine, they have their own cell surface receptors. Glucagon isn't binding to the beta-adrenergic, and epinephrine isn't binding to the glucagon. They have their own receptor, but they sort of do the same thing. And glucagon, when it says glucagon, liver. Epinephrine, muscle and liver. The muscle is much more sensitive to epinephrine. The liver is much more sensitive to glucagon. But glucagon isn't just a liver thing. This stuff is all over the place. It's in the kidney, these receptors, right? No, it's very prominently featured right in the liver, but you'll see it in the kidney, the heart, you'll see it in the GI tract, see it in fat. Glucagon signaling is going to be all over the place, and same thing, epinephrine is going to be all over the place. But this signaling cascade, right? This is the G protein coupled receptor. There's adenylyl cyclase, again, adenylyl or adenylyl cyclase. Let's convert ATP into cyclic AMP, and cyclic AMP binds to PKA, protein kinase A. It's a kinase. It phosphorylates stuff. Let's phosphorylate phosphorylase kinase and glycogen synthase. Let's turn this one off. Let's turn off glycogen synthase. We're being catabolic here. So you phosphorylate this one, it shuts it off. You phosphorylate this one, it turns it on. And what does this one do? Phosphorylates this one, glycogen phosphorylase. What does that one do? So it's chopping off little G1P chunks off of your glycogen. We've known this for a long time, right? So Sutherland, this is work from the 50s, right? So this isn't new. Cell signaling cascades. It's not like that weird new thing. mTOR understanding is very new, but 1971 Nobel Prize went to Sutherland for his work in the 50s on catabolic signaling, on mobilizing glycogen in the form of G1P, and then phosphoglucomutase converts into G6P, and then whatever you have glycolysis to go. Now, protein kinase A, it phosphorylates a lot of stuff. This kinase doesn't just like I have one, act. It's not like the NFL where like I'm the guy who kicks. I'm the guy who stops you from tackling the quarterback. I don't know the names of any positions. Kicker, what I can do, and quarterback, I can do that one. Uh, but like everyone in the NFL has very specific things. I'm the guy who goes out for passes, right? I'm the guy who tries to kick a field goal. People have very specific. PKA does not work like that. That's not what PKA. Maybe hormone sensitive lipase. That characterizes it. But protein kinase A, protein kinase B, these things, they're sort of well controlled loose cannons. So the idea of this loose cannon, it just goes anywhere. You can't predict what that thing's going to do. Super well controlled, can go anywhere. These protein kinase enzymes, and phosphorylating pyrrolipin to expose that lipid droplet to allow lipolysis to take place, to allow those lipolytic enzymes to do their business, do their chopping. Now, why do we have to do that? Now, again, this diagram's a little bit long. A lot of these diagrams, people sort of miss the point a little bit. Hormone-sensitive lipase, right here, that one is doing this. Diacyl glycerol, monoacyl glycerol, so that's monoacyl glycerol lipase. The triglyceride here, this is adipose triglyceride lipase, is a different, but PKA, hormone-sensitive lipase, this one. Why do we need to go through lipolysis at all? Why can't we just burn fat? Why do we have to get these little fatty acids, get these like acyl CoA? Why do we need to do this? Well, this is huge. The log is huge. Your fireplace. You can't just throw a log in the fireplace, maybe a beach fire or something, but in your fireplace, little like hearth and all that, you have to chop up the wood first, and then you can burn it. It becomes small enough to burn. Like you can't light this on fire. It's not even going to fit in there. It's ridiculous. You have to chop it up first. That's what lipolysis is. Chop it up into its little parts, and it's cell signaling cascades that do it. Cell signaling cascades do all of that chopping. Now, AMP and cyclic AMP, two different things here, right? The second messenger the Sutherland discovered in the 50s, right? This is cyclic AMP. AMP is not activating PKA. When like I don't know, a Capricorn and an Aries or something, whatever. Maybe they aren't compatible. That's like AMP and PKA. They're like a Capricorn and an Aries. But let's say like a Sagittarius and a Libra are super compatible. That's cyclic AMP and PKA. I know nothing about um, any of the astrological anything. So I may have just gotten everything backwards, but you get the idea. So AMP, PKA, they're not friendly, right? They're not going to hook up at the discotheque. Cyclic AMP though, this and PKA, good luck keeping them apart. And so then we have adenylate cyclase, AC. Adenylate cyclase is going to convert your ATP, this guy, into your cyclic AMP, this guy, right? So ATP becomes cyclic AMP. Cyclic AMP is what binds to PKA and turns it on. Now, once you have your cyclic AMP, you know you can just activate cyclic nucleotide phosphodiesterase. This is just PDE, phosphodiesterase. There's a bunch of phosphodiesterases. 3B, that's the one that you're going to see in fat. And um, if you have caffeine, right, is an inhibitor of phosphodiesterase of PDE, a non-specific one. Um, there's so any of the methylxanthines. These are going to be inhibitors of your phosphodiesterase, and caffeine is just one of them. But you will convert your cyclic AMP into regular AMP, and that's not going to activate PKA. So our phosphodiesterase is what does that conversion, which blocks PKA. We block PKA activation with phosphodiesterase. Once we've built up the cyclic AMP, that's how we stop it. So the narrative version of this, insulin binds to its receptor, being this prominent PDE activator. Insulin binds to its receptor, its insulin receptor. Some stuff happens. PI3K, phosphatidylinositol 3 kinase, gets activated. Some stuff happens. PKB gets activated. Protein kinase B, seriously loose cannon and seriously regulated. As long as if it's unregulated, the disease states manifest in a hurry. PKB phosphorylates PDE, phosphodiesterase. PKB is a kinase. It phosphorylates stuff. It phosphorylates phosphodiesterase. And also PI3K. There's evidence that PI3K does it too. There's PI3K and PKB are activating phosphodiesterase. There's experiments where they block PKB and they still see PDE activation. But if you take wartmenin, which is a PI3K inhibitor, then you don't get phosphodiesterase activation. So PI3K, PKB, we're both activating PDE. PDE converts cyclic AMP into AMP. So, 4 of these little cyclic AMPs should be binding to PKA to activate it. That doesn't happen because you break them down. And so you don't activate hormone sensitive lipase. You don't phosphorylate activating hormone sensitive lipase or pyrrolipin. And so lipolysis doesn't happen. And you can say the same thing with any of these cascades. This is one of the best illustrations, and pretty much everything on here so far. Um, so this is if we're going to have epinephrine bind, it's a beta-adrenergic receptor. We activate adenylate cyclase. Adenylate cyclase converts ATP into cyclic AMP. Cyclic AMP binds to PKA. PKA kinase phosphorylates pyrrolipin and hormone-sensitive lipase. You see a little P that's phosphorylating, and those are responsible. These sort of these primary regulators, the acute regulators of lipolysis. So this whole cascade. We've gone over this. Insulin binds to an insulin receptor. That's insulin receptor substrate. Here we see PI3K, phosphatidylinositol 3 kinase again. And what that does is technically convert PIP2 into PIP3. And so PIP2, phosphatidylinositol bisphosphate. I don't care if you say diphosphate, biphosphate. It's the same thing. You know what I mean? There's 2 phosphates. Phosphatidylinositol, 2 phosphates. Who cares? And so you get to PIP3. And PKB, technically the activation of PKB is phosphoinositide dependent kinase, but we'll talk about that. We'll get there. But you need to go through this PI3K step to get PIP2 to PIP3. And now we get PKB activated. PKB, we are activating phosphodiesterase, PDE3B, right? So we're in fat here. This is lipolysis. This is a fat cell. So that's the cell wall, right? And we're inside of the cell over here. Out here is outside of the cell, which is where your epinephrine is, your insulin is outside. And here we're inside of the cell. But if you activate the PKB, the PI3K PKB pathway, you are turning on PDE and you're inhibiting this pathway. So anabolism predominates. I don't care if you bind a little epinephrine, have a bunch of insulin there, and that's powerful, right? You're breaking down, you're halting this cascade. You're not halting this cascade with this one. You're halting this cascade with this one. So it sort of has, like the dominant cat that goes and like pees all over the yard and says mine, mine, mine. That's sort of anabolism behaves in that way a little bit. Now, catabolism behaves in that way with AMPK, and we'll get there, but more on the insulin, on insulin signaling. So insulin receptor substrate, your PI3K, your PDK. Again, we'll talk a little bit more about that one. We're missing PIP2 to PIP3 here, whatever. But and then you get to PKB, right? And PKB, glycogen synthase kinase. This again, we should probably change some of these arrows. I really don't like this one that much, but what it shows is because also like this arrow is that it doesn't do that, right? To mTOR, but we'll talk about some of these other arrows later more accurately. But PKB, we are mobilizing GLUT4 to the surface, GLUT4 translocation, and so that is an anabolic. Let's get some sugar into the cell, but then through this cascade, we're going to also turn on glycogen synthase. Right, we're going to activate the synthesis of glycogen. So this is the core of insulin signaling. Is really this PI3K, PDK, PKB. And it's the same thing. Let's look at a different diagram. It's the same thing. AKT is PKB, but we add another step here. Tuberous sclerosis complex, or tuberin. Just write tuberin, which is technically TSC2, but it's a complex. This is a protein complex, and TSC2. This one is tuberin, and PKB is phosphorylating, deactivating tuberin. Now, tuberin. This is a major hub for interaction with mTOR. Tuberin is one of these major hubs where stuff interacts with mTOR, not just PKB, other stuff too, and we'll get to that. But so tuberin, technically what this is doing, there's a missing piece right here. Tuberin is shutting off a thing called RHEB, R-H-E-B, Ross homolog enriched in brain. Who cares? R-H-E-B, RHEB. Tuberin is shutting off RHEB, and RHEB is activating mTOR. So it gets a little bit confusing because PKB shuts off the thing that's shutting off the thing, RHEB, that turns on mTOR. So there's a double negative. PKB shuts off the thing that shuts off the thing. It's flip the light switch twice. Okay, so if it was already on, you shut it off, whatever. So you get the idea. And then downstream from mTOR, there's P70SSK. I've referenced that before. And you've seen 4EBP1 before, but protein synthesis. Let's get to that ribosome, link together a bunch of amino acids, synthesize some proteins. Let's do some translation. Let's build us some proteins. P70SSK. There's another step here. There's ribosomal protein S6, RPS6. But then 4EBP1 is shutting off protein synthesis. P70SSK is turning on protein synthesis. mTOR is a kinase. It's a phosphorylating enzyme. So mTOR phosphorylates both of these. It's going to phosphorylate P70SSK to turn on protein translation, protein synthesis. Let's make us some proteins. And it's going to phosphorylate, shutting off 4EBP1. Remember in PKA signaling, how there are multiple things phosphorylated. We get glycogen phosphorylase phosphorylated, which turns it on. So we're going to chop up some glycogen. We get glycogen synthase phosphorylated, which turns it off. So let's stop putting glycogen away. You shut off the thing you need to shut off and turn on the thing you need to turn on. You don't just flip on the light switch, right? You I don't know, that's a bad example, but you have to shut off inhibition and turn on promotion at the same time. And there's a lot of this sort of overlap in the body at various places in the body. You're going to see this overlap, and mTOR is one of those where it's a kinase and it's going to phosphorylate the thing that turns on protein synthesis, and it's going to phosphorylate turning off the thing that is restricting protein synthesis. So there's a couple of pathways it's going to do. So when we expand the diagram, you can see there's a bunch of additional activity of PKB. PKB goes all over the place. As earlier in this lecture, I said it's like pan kinase B, right? Protein kinase B, but it's like panoramic, pan like pancreas, all flesh, like it goes everywhere, right? This protein kinase B really just goes all over the place, and there's a lot. You see the insulin receptor substrate. You see PI3K. The opposite here is going to convert your PIP3 into PIP2. But then you see your PDK, and then activation of PKB. Again, we know this. PKB is inhibiting tuberin. Tuberin is actually inhibiting Reb, which isn't here. Reb turns on mTOR. We know what's downstream. But there's a bunch of other stuff too that PKB is doing. And we could go even further. We're not going to go that much further. We're not going to cover all this stuff, but you would start to recognize many of these things. Growth factors, you see insulin receptor substrate there, integrins, PIP2, PIP3, PI3K, and P10. You see cytokines up here. If you see JAK-STAT signaling, there's a lot of these things. When you see RTK, receptor tyrosine kinase, this is just, you don't need to know that, just types of receptors. You're going to encounter that a lot though. RTK, you'll keep encountering that. But you see a lot of things that are beginning to look familiar. And really some of the major domains of PKB, major domains are proliferation and growth. So these two go together. You're not going to proliferate before you grow, right? You gotta grow these things a bit first. You can grow these cells and then we can divide them. Let's get enough territory and then we can divide this, right? So you're going to see a lot of this growth first. And we talked about tuberin, tuberous sclerosis complex 2, tuberin. Pras40, we'll get to this one, proline rich PR, AKT substrate. AKT is PKB. So, and 40, that's just his molecular weight, is 40 kilodaltons. And so we'll talk about that one in a little bit. You can figure out exactly what this is based on get rid of proline rich, AS40, AS160. I'm sure some of you have guessed in your head what that stands for. That's just AKT substrate, and the molecular weight is 160 kilodaltons, right? So this is the thing that is restricting GLUT4 translocation, and so it's inhibiting the motion of GLUT4. And so if PKB shuts off this thing, this substrate, it shuts off this thing, then you can mobilize your GLUT4. So that's actually how the GLUT4 is being mobilized. So survival, when we start looking at pro-apoptotic things, apoptosis, this programmed cell death, we get like BLC associated agonist of cell death. So B, blah, 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 agonist of death. That's what bad is. It's B, agonist of death. And so this is we're getting into apoptosis and cell death. And cysteine, aspartic protease. A protease is we're breaking down proteins, right? So we talked about proteases with like neutrophils when we were in the immune system and what they're doing. So this is like we're killing stuff over here, and PKB is shutting off that killing stuff, section, and PKB is shutting off the thing that's shutting off glucose uptake. So we turn on glucose uptake, we turn off proteases, and we turn off apoptosis, and we have a lot of metabolic things, but metabolism, as you know, is a big subject, the sum of all chemical reactions, and proliferation. So PKB does a lot of things in the body. Now, FOXO, the PKB FOXO switch in aging and cancer. FOXO, fork head box O, these are transcription factors. Autophagy, autophagy. So, like, what does a macrophage do? A phagocytic cell, sort of ingestion, autophagy, autophagy. This is what we're looking at with FOXO. AMPK is going to activate these things, and that's wonderful if you have cancer, right? This is you want a bunch of FOXO activity. PKB inhibits it. Now this is great in terms of warding off atrophy. Get this FOXO switch going, and yeah, this is bad for cancer, but um, if you inhibit FOXO, it's bad for cancer, but it's great for hypertrophy, right? Because you're shutting off, you're minimizing atrophy with PKB. AMPK is activating this thing, which is terrible for muscle metabolism if you're looking at growing. Now, why do we have cell signaling cascades? Like, what is the purpose of these things? We're back to why questions. Remember, why is the sky blue? Purpose, I don't know how to answer that one. So we're not doing how right now, we're doing why. Why are cell signaling cascades important? We need information to be relayed into our interiors, into the insides of our cells. The insides of our cells, we use weather apps. If you want to know, you just like Siri or whatever, and you ask, uh what is the weather today, and then you plan your schedule, you plan your attire or your activities accordingly, right? So we have some meteorologist somewhere doing predictions, using the computer and relaying that message to somebody else who programs it into some app, and somebody they're signal. I mean, this is a relay race to get the weather information to you. You didn't talk to the meteorologist, right? You didn't interpret the weather yourself. You certainly didn't interpret the weather yourself. You didn't go out and and measure a bunch of things over the course of the week. And there are relays of information throughout our own lives, and so it actually makes sense in the body would do this, because it's how we behave too. So we need to know what the weather's like outside, environmental threats and stresses. And we need that information to be relayed to us to make up our decisions. Our body, what is the environment out there? Is it very acidic? Is it our tissues breaking down? So think about like prostaglandins. What are prostaglandins doing? It is a marker of tissue damage. A bunch of arachidonic acid, your tissues are getting banged up, and this arachidonic acid is being released and converted into prostaglandins through cyclooxygenase. You know that. And now we have this marker of tissue damage. You have a ton of prostaglandins, you must have a ton of tissue damage, right? And so let's relay that information to our cells. Let's bind to the cell surface and say, hello in there, just letting you know, shit's bad out here. We're really banged up, so please, get to business, repairing and remodeling, and hopefully reinforcing, make it so this same exposure won't happen again. It's a little bit like wind chimes. You hear the wind chimes outside. The wind chimes don't walk inside and tell you it's really windy out there, I'm just letting you know. The wind chimes just, they're outside, and you hear them blowing, like, okay, I know what the wind is doing. Cell signaling cascades, it's very similar. That's how cell signaling cascades work. Now, the concept of upstream and downstream variables. If something is upstream, think about, you eat a hamburger. Okay, it has a bunch of bread in it. Bread is super high glycemic, and you're going to get an insulin response. The beta cells in your pancreas and your everywhere, your flesh all over the place in your pancreas are going to release insulin. Insulin downstream. So the ultimate upstream is a triggering event. It's, I ate a hamburger. Downstream from that, blood sugar goes up. Downstream from that, pancreas releases insulin. Downstream from that, insulin binds to a cell surface receptor. Downstream from that, PI3K and PKB, and downstream from that, mTOR, and downstream from that, protein translation, right? And GLUT4 translocation, all stuff. So that's that concept of upstream and downstream. And then you could also say there's protein in here, and in that protein, there's leucine, there's lysine, there's arginine, there's stuff that's going to trigger mTOR in a different way. So there's upstream, downstream. If you go exercise, let's the example we just gave. You go exercise, you go garbage up a bunch of sarcolemmas, right? A bunch of muscle membranes. And in that phospholipid bilayer, there's a lot of of arachidonic acid, and it gets released during all this exercise. PLA2 is releasing it during all this exercise, gets converted. Cox converts it to prostaglandins, and so this downstream, 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 and then prostaglandins bind to their receptor. Downstream from that, we see MAPK, mitogen activated protein kinase cell signaling happening. Downstream from that, we see crosstalk interaction with mTOR. Downstream from that, we see protein translation. So upstream, the ultimate upstream, the very first initiating event, right? Downstream from that, we have whatever physiological phenomena. Downstream from that, we have the next step. If it's a phosphorylation cascade, what did it phosphorylate? So PKB, what's it phosphorylating? PKA, what's it phosphorylating? PKA phosphorylating phosphorylase kinase, is it phosphorylating glycogen synthase? Is PKA phosphorylating hormone sensitive lipase? Is it a pyrrolipid? That's downstream. So that's when I talk about upstream and downstream. That's what I'm referring to. Now, again, cells get busy. Cells get busy. There's so many cell surface receptors on these things. So this is a cell, this orange thing. This is the, no, your cell is this like perfect rounded, sort of like social networking post rounded rectangle. But that's your phospholipid bilayer, right? That's the wall on the outside of your cell. A cell surface receptor are all these things. So you see that RTK again. We encountered that one, that receptor tyrosine kinase, the RTK. And over here you see integrins. You know all about integrins. Um, and so we're integrating that extracellular matrix, what's going on outside of the cell. We are integrating that with the interior of the cell, a transmembrane protein. Earlier, I mentioned focal adhesion kinase, FAK. That's its sort of tag team partner that's going to relay a lot of that message. But so let's just go through some of these. JAK-STAT. We've talked about JAK-STAT. Genus kinase over here. This isn't the same thing. This is a second messenger system, but it's not the same second messenger system as cyclic AMP, PKA, or going through PKB. It's its own. It is its own thing. The growth hormone goes through that. There's some cytokines. You're going to do that one. We're going to do JAK-STAT. Up here, so you see the PI3K, PKB, and getting into a lot of mTOR signaling from there, and so that's what or a G protein coupled receptor, and so you'll see adenylate cyclase down there, and whatever. So there's all these growth factors and endothelial growth factor, transforming growth factor, IGF-1, insulin-like growth, all these different growth factors, and insulin would be up here. But all this stuff that's going to do a lot of PI3K, PKB, mTOR signaling. Integrins. You are familiar with them, but we're going to talk about them in real detail during this block. So the integrins that are relaying their message, that's mechanotransduction. Now you'll see from FAK, just follow the arrows, and you will see PI3K activation. That's one of the ways it works. One of the ways that integrins work is through PI3K signaling, but it's not the only way. Insulin has to go through that pathway. Integrins don't. Integrins don't have to go through that pathway. Wnt, the Wnt proteins, WNT, it's pronounced like WINT. Wnt, they bind to frizzled, right, and then go through disheveled, and that's where you see glycogen synthase kinase again. You've seen that one before. There is a relationship with the Wnt proteins and mTOR. There's also a relationship with autophagy, but more interestingly, Wnt signaling, those proteins, they will deactivate tuberin. 15 minutes, thanks. Wnt proteins will deactivate tuberin, and the way that they do that is inhibiting glycogen synthase kinase, GSK3 specifically, but glycogen synthase kinase. Now, the PKB has this effect too. It's sort of a doubling up. I'll show you that effect of PKB. But if you inhibit glycogen synthase kinase, that thing is promoting tuberin, glycogen synthase kinase, promoting tuberin. If you turn that thing off, you stop promoting tuberin. And remember, tuberin is inhibiting REB, RHEB. REB is the thing that directly activates mTOR. So GSK, glycogen synthase kinase, is promoting tuberin. the tuberous sclerosis complex, and it's directly phosphorylating. It's a kinase, glycogen synthase kinase. It's phosphorylating tuberin, TSC2. And if if you can shut off GSK, you can turn on mTOR. If you can shut off tuberin, Reb is free to turn on mTOR. So the Wnt proteins do have an interaction with mTOR. And then down here, you see hedgehog. I'm not going to ask about any hedgehog thing, but literally sonic hedgehog is the name of one of the ligands. And desert hedgehog and Indian hedgehog. You can tell the type of people who do the chemistry, pharmacologists and chemists and maybe biophysicists here and there who are discovering and identifying and being able to name these things are also super nerds. And so Sonic Hedgehog is obviously named after Sega. There's a lot of embryonic roles here. I'm not going to ask about the hedgehog stuff, but teeth, stuff like that, embryonic roles. But in adults, there's some proliferation of stem cells. So there are roles in adults, but it's on this diagram, and so why not mention it? Down here, death factors. Tumor necrosis factor, whatever, death factors. This is just programmed cell death. And so if we are killing off our cells, that's all that really is. And we're not going to do too much talk here. We'll do a little bit, but for the most part, we're going to be up here with these ones, the integrins and the PI3K, PKB signaling. And I'll mention the Wnt proteins a couple additional times. But the hub, the core, the primary little section of mTOR is PI3K, PKB, and then PKB goes all over the place. So we talked about FOXO, right? Fork head box O, that we're inhibiting FOXO. PKB is also inhibiting glycogen synthase kinase 3. And that's the thing that the Wnt proteins would also inhibit. And that's turning on tuberin. And tuberin is inhibiting Reb. You see Reb there. Now, Reb activates, that's the direct activator of mTOR, mammalian target of rapamycin, mTOR. And there's some other core players in that complex. mTOR is a complex. So I mentioned mTOR complex 1 is our hypertrophy. mTOR complex 2, there's a lot of cell survival stuff. We'll talk about mTOR complex 2, but this is mTOR complex 1. So when you see mTOR C1, that's mTOR complex 1. Remember when we were doing like AMPA and NMDA receptors, and you see like NMDAR, NMDAR, right? You'd see like that. That just means receptor. It just means NMDA receptor when you see the R. When you see mTOR C1, that's just mTOR complex 1, which is this. It's a complex. There are a bunch of proteins here. Deptor and MLST8. Sometimes this is called Gable. Usually when people are speaking, they'll call it Gable. G protein beta subunit like G beta L Gable. And so Gable is easier to say than MLST8, but when people write it, usually they write MLST8. Press 40, Deptor, Raptor. Now, Raptor, this is, earlier I said TSC1 and 2, tuberin, really critical interaction point with mTOR. And you're starting to see it already with like GSK, with tuberin, but lots of stuff is going to interact at the tuberin step. Raptor, another critical site of interaction. And you can phosphorylate this on multiple sites and help turn it on, turn it off. And Raptor, regulatory associated protein of TOR, of mTOR, right, of target of rapamycin. So mTOR is a kinase. Raptor is this critical protein. If you don't have a raptor, you die. You don't have this, cell signaling. Now, mTOR has to recruit its downstream target somehow, right? This is P70 S6K. Here it's S6K. Sometimes it's S6K1. Whatever. There's a lot of names for this thing. Remember, this is the thing that turns on protein synthesis. And there is ribosomal protein S6, RPS6 is almost always how it's written. The most common way that P70S6K is written is P70S6K. The most common way this is written is RPS6, ribosomal protein S6. And over here, 4EBP1, right? This is the thing that's inhibiting translation. This is halting, policing protein translation, protein synthesis. Over here, we're turning on protein synthesis. So mTOR is a kinase that phosphorylates. You see these little phosphates over here. mTOR phosphorylates P70S6K and 4EBP1, shutting off this one and turning on this one. Now, this is a kinase too, right? So it's going to phosphorylate ribosomal protein S6. What do you think that interacts with? Ribosomal protein S6, right? The ribosome. That's where translation happens at the ribosome. That's the kitchen where you cook up your meat, right? Where you cook up your proteins is the ribosome. So this is mTOR, really sort of the core hub here. Now, Raptor, this main function, I'm going to talk about a lot of Raptor things, but its main function is recruiting the downstream targets. It's a headhunter. It recruits the downstream targets so that mTOR can phosphorylate them. So the mTOR can't just be like, all right, it's not like a sharpshooter. You have to you have to recruit these things. And for mTOR to recruit them, Raptors is necessary. Now, the finish line of a lot of this stuff, often the finish line of cell signaling cascades is the nucleus. It doesn't have to be, right? It doesn't have to be. It could be the ribosome. It could be GLUT4. I mean, there's a lot of things it could be that aren't nucleus, but often the finish line is nucleic. And transcription and translation, just reminders of what those things are. Transcription, right? Let's go make our mRNA. And so our sort of genetic information, our recipe, let's make a recipe and let's send that recipe to the kitchen, right? Let's send that recipe to the kitchen where we can make a protein. We can link together a bunch of amino acids in a very specific order, and it folds up and does its behavior based on its shape. And so that is the ribosome is the kitchen where you send the recipe to, and you cook up your proteins there. You link together all those amino acids, hopefully doing it correctly. So this is the slide we'll end with today. This is our checkpoint. If you ever play like old racing games, this is Rad Racer. There's just like, if you don't hit the checkpoint, you sort of coast and then you die. And then like you hit the checkpoint and suddenly you're gassed up again, you can keep going. It's like the arcade version, so you can keep eating people's quarters. Put more quarters in, you keep going. So this is our checkpoint. This is sort of where we'll stop today. So what is mTOR? mTOR, over here we have complex 1. Over here we have complex 2, but mTOR is a kinase. It is a critical regulator of protein turnover. It's a protein. It's an enzyme. It's a kinase, a phosphorylating enzyme, but it is this major hub in your cells that regulates growth states, right? Are you growing or are you shrinking? Is it hypertrophy or atrophy? Where are you? Protein metabolism, protein turnover, and synthesis degradation. mTOR is this massive hub for that. And there are two different complexes. How many different mTOR complexes are? Well, there's two like described ones, and maybe there's a third, whatever. But there's two of these hubs, and you can see Richter, rapamycin insensitive. That's what the RI stands for. Companion of TOR, of it still ends in TOR. So Raptor is this regulatory associated protein of TOR. And so mTOR, mammalian target of rapamycin, right? Richter, rapamycin insensitive. So that sort of telegraphs our paths of where we're going from here. Is we're going to start talking about some of these other regulatory proteins and how to interact with them. But how does mTOR signaling work? It's phosphorylation cascade, right? You have an upstream activator, and there's going to be different activators of each. Technically, mTOR complex 2 is an activator of mTOR complex 1, but mTOR complex 1, this is our hypertrophy. And and so I'll talk about the downstream targets, the upstream targets, but mTOR signaling is regulation. It's a massive metabolic regulator, and um, you downstream from complex one is just a ton of hypertrophy. Let's make cells grow. Let's translate proteins. Okay, what questions do we have?
I have a question. I was gonna ask, do you know of any good? It's kind of maybe a dumb question. Do you know of any good YouTube channels or anything that can like help explain this further, especially since we're online, just so like, we can really understand these concepts? 'Cause, I mean, we will rewatch these lectures, but um, I don't know. Is there anything that you recommend in ways of studying this virtually?
Yeah, quite honestly, the researchers themselves don't just go get like, don't get like some peer. Like I'm sure there's videos where it's just like somebody taking a class and they're trying to explain it. And those are always wrong. Go to the actual physiologist. Go to. I mean, we're talking like David Sabatini. He's the best one. And even if you look at the original names of these things, it wasn't always called mTOR. It had a few different names. Raft1 was David Sabatini. FRAP, FRAP, that was Stuart Schreiber. And but these names, these researchers, the person I think is most articulate, most knowledgeable, and most available is David Sabatini. So look him up, and he'll have videos. You'll find something by him, and he'll talk about stuff. Really articulate guy, and he has done like half of the landmark studies in how mTOR works. It's his actual study, so he's not just like some guy like at the gym who's like, I heard about mTOR, let me try to tell you about it. He's a foundational researcher who his entire career is mTOR, and he sort of like he pretty much discovered it. I mean, it's really there's several founding names of who the major contributors are to mTOR. This guy Robert Abraham is the one who actually named it mTOR, but FRAP or RAFT1, RAFT1. So if you see stuff like that, that's about mTOR too. Those are just alternative names for it. At some point, it won't even, like, people have started dropping the M. So mTOR, mammalian target of rapamycin. Um, at first, people changed the M and said mechanistic target of rapamycin. And I like rolled my eyes and ears and whatever. I rolled everything. I did somersaults with like disapproval, because it's just stupid to change the M like mechanistic target. This is ridiculous. And so if it's mTOR, it's mammalian target of rapamycin. I found mechanistic just to be the stupidest alteration. And then some people started dropping the M, which I think is fine. Target of rapamycin, totally fine. Just drop the M, because at some point we're actually going to drop the TOR, because that's sort of stupid too. It's this critical enzyme for survival and metabolism and the balance of hypertrophy and atrophy. I mean, this thing, if it gets unregulated, good luck staying out of the coffin. And do you want to grow? You better get mTOR going. It's not going to work otherwise. And this thing is so important to us. And we call it target of rapamycin. And rapamycin is just like this exogenous thing. Like, I guess it's like calling, um, I don't know. It's like if there's like PKB, let's say this is PKB, protein kinase B. You saw how many arrows are going all over the place. I mean, it looks like fur. It looks like fur coming off of it. And if you called PKB target of insulin, that's stupid, right? That's not like, and that's actually better than calling mTOR target of rapamycin, because rapamycin is like this exogenous thing. Insulin, at least we're producing that ourselves. But target of insulin. Well, a lot of stuff. This is a PKB is a critical site. Yeah, it's target of insulin. And so that's mTOR is actually a stupid name. But it's actually, it was a better name than FRAP and RAFT1. But if you see in the literature or in videos or whatever you go look for, FRAP or RAFT1, that's the same thing. But yeah, David Sabatini is the person I would recommend. He's the best mTOR researcher alive, and who ever lived. He's like the guy for this.
Thank you. And then
Yeah.
one quick thing, I know other people have questions too. One second question I wanted to ask really quickly, and I know we'll get into it, is what do you think the number one mistake that people make when they're trying to activate hypertrophy is? Is it eating food? Is it not taking in lysine, arginine, a complete protein, like that conversation you, Vanessa, and I had last semester of like just whole proteins and stuff like that? What do you think is the number one mistake?
You know where people talk about a balanced diet? I have a balanced diet. I have the right, macronutrients and the right micronutrients. I have all my food groups and I have all my, I have a balanced diet. I think you need mTOR itself sort of needs a balanced diet. It needs a balanced input of you don't want to just overdo one thing. It's like, well, I mean, even like, I don't really eat carbohydrate. I mean, it's, I eat broccoli and like cauliflower and stuff, but that's it for carbs. And that's not helpful for mTOR, and I'll get to those reasons why, the reasons how rather it is not helpful. And but as you go through, PI3K, PKB signaling, and as the nutrition from the protein end, and from the carbohydrate end, if you're getting in the right amino acids, yeah, I've mentioned this leucine, lysine, arginine is like 90% of recognition of the protein, but so you want all the different inputs. You want sort of a balanced diet for mTOR where it has sort of the insulin, PI3K, PKB inputs. You have the sustenance, right? The availability of amino acids. You have to have that too. And you want some mechanical loading in there. I mean, you want to get all of these different inputs. And so we'll end at the beginning when I said like the 5 steps that we're going to go through. The 5th step is, let's talk about specificity of adaptation, and let's talk about a little bit of biomechanics and Henneman size principle, and all of these concepts that we've discussed earlier as ingredients into mTOR signaling, and how to balance the diet optimally. So I promise we'll get there. It might be like, another 4 lectures or something, maybe 3 or
Thank
4
you.
lectures from now.
Awesome.
Other questions?
Yeah, just mulling over everything going on. So like, it's a lot to just take in.
Oh, yeah, no, it's a ton. Yeah, I actually don't expect people to have. Jay, your question was great. Yeah, where do I go for resources to learn more? That's a great question. And uh, yeah, so there's so much new vocabulary and so much new information, and it's going to be this way for the next couple of days. And then when I get back, so right now it's just, and I'll upload this immediately so you can study from this immediately. Like, you just give me 15 minutes after class, 20 minutes, whatever, to get it uploaded. But the next few lectures, it's just like trudging through the swamp of like, what is all this new stuff. I mean, this is like, you signed up for German and you just get plopped into German 4 or something where nobody speaks any English, and like, I don't know how to follow. And so it's going to feel a little bit like that probably for the next couple of lectures. And then when I start rehashing it should all click if you study along the way, and if you learn the vocabulary, and rewatch some lectures and write some stuff down and think about it. Everything will click, and it's not that bad. But yeah, at the first blush, it blushes with some heat.
Yeah, it's a lot to digest. Yeah.
Yeah, it will. You'll sleep on the calories, and you'll put them into circulation. But that's if you go to the buffet and just you put in 8,000 calories, I mean, just lie in bed with your aching stomach. They'll find their way into circulation, and you can put those calories to use later. So that's sort of what we're doing now, is I'm just cramming words and sort of physiology into your mouth, and like it'll find its way to circulation. You just got to give it a little bit of time, and chew it extra hard. And once it clicks, mTOR is great. And so the people who have come out of this class and sort of loved the content and employed it, mTOR is the thing they talk about. And it's sort of it is the the peak of the class. If there's like there's a few subjects that I really like, and that probably comes through in my energy when I teach. Although I don't have much energy today, but it's just like darker than it normally is. And so I think that's sapping me a little bit. But size principle, orderly recruitment size principle, specificity of adaptation, and mTOR. Those are really that's my, trinity, I guess, of physiology. So yeah. No other questions for the day?
Yeah, I actually had one. Can
Okay.
you like microdose epinephrine as a way to supplement weight loss? Because like, doesn't that kind of lead towards lipolysis?
Yeah. People will do beta agonists, and when we get to the supplement section, I talk about drugs too, because what's the difference? So yeah, you can. Clenbuterol that's what people will do, is you clen, and you get shaky a little bit, and you actually can. It's fascinating. People get some hypertrophy off of it too. I'll get there. We'll talk about that. But yeah, a beta agonist, you can do the pill forms of beta agonists, not like inhaler, whatever, not like doing inhalers, but that's not gonna work in the same way. But pill forms of beta agonists, you can do that.
Yeah, I was just wondering because it sounds like epinephrine or norepinephrine would be one hell of a pre-workout, Kind of get a
Yeah.
little.
Yeah, go do clen and get into In Shape, or whatever. Is In Shape going to go out of business? All these, are they going to stick around? The gym?
They've already closed like 7 locations, I think.
Have they really? Is the Stockton one still open?
One of them closed in Stockton. The Lathrop one closed, and several other ones have closed down.
I think a lot of 24-hour fitnesses have closed too.
Yeah, I think 24 Hour Fitness like nationally just went bankrupt.
Yeah, the 24 in Stockton closed a couple months ago.
Okay. But yeah, so whenever you can get back in the gym, if you're doing some like beta agonist, you'll feel it. That's a much better pre-workout than caffeine works well. Caffeine's effective. Theobromine, that's the one in chocolate. Just have some. People talk about like, oh, there's caffeine in chocolate. Yeah, okay, there's some caffeine in chocolate, but theobromine is another methylxanthine that's there. Or take theophylline. That's the prescription that you could get of theophylline that's been around forever. So all these methylxanthines that are going to have a PDE inhibition effect, but if you just take a beta agonist, that's going to be even more effective.
What do you think about cranberry juice for pre-workout? Because I know Eddie Hall always talks about it, and kind of goes off. And it seems like a really weird pre-workout. But do you know chemically why that would make any sense, or is that just an anecdotal opinion?
I'm unfamiliar. It doesn't mean there isn't something there. It's just I have not encountered literature on it. People have been doing for the last few years like beet juice, like beetroot juice, and maybe 3 or 4 ACSMs ago. I think 4 ACSMs ago, it was just there was like 100 posters on using beets, as an ergogenic aid for cardiovascular effects. There seemed to be some effect for that, but and before that, I had never encountered the idea of Dwight Schrute supplements. So just because I don't know something doesn't mean it's not there. It's not real. There's no effect. I have not encountered anything with cranberry juice though, unless it's like my like urinary infection is really getting in the way of my workout. Okay, all right. I have heard that. I have heard an effective use of it in that capacity. I really have to shave this thing. It itches. Okay. I will see everybody on Friday.
Yeah.
We're going to begin in the middle of mTOR. Right? I might rewind it a couple of slides and then just to get our footing. All right, here's exactly where we were. Let's start running forward. So we'll just pick up where we left off essentially. See everyone.
All right. See ya.